Hi, my name is Scott Banta. I run the Protein and Metabolic Engineering Laboratory at Columbia University, and I'd like to tell you a little bit more about our work. In our lab, we spend a lot of time engineering proteins for different applications. Protein engineering provides a set of tools and techniques so that we can engineer protein molecules at the atomic level. We can make changes to amino acids within the molecule, and then like to do is to explore how these changes affect the molecular behavior of the molecules. We can look at catalysis, ligand binding, changes to structure. We can then move on to how this affects then more bulk behaviors. So we can look at how this affects self-assembly, we can look at how this affects design materials and different structures. And then finally, we spend a lot of time in my lab looking at how this then affects final applications, ranging from biomaterials to bioelectronic devices, biomedical applications, and all sorts of things. And ideally, what we'd like to do is to be able to relate this back to how we were able to make changes in the protein structure, how that can impact the final downstream application. So in this video, I'll just tell you a couple of different vignette stories that we've had through the years. These are some busy slides, but just to give you kind of overview of some of what we do. First story I'll tell you about is a work that we've done with this enzyme here. This is a alcohol dehydrogenase that comes from an extremophile bacterium that's able to survive boiling water. So this protein is able to therefore survive extreme temperatures up to 100 degrees C. And we've been able to do a lot of things with this protein over the year. So we've been able to engineer it for self-assembly so that it forms these hydrogel biomaterials. We've done a lot of work trying to understand its kinetic mechanism. The enzyme uses a cofactor, NAD. We've been able to engineer the enzyme's ability to use that cofactor and to change it to use another cofactor, NADP. Along the way, we were also able to engineer it to use a minimal cofactor. This is nicotinamide mononucleotide, and explored how using the minimal cofactor improves the performance of biofuel cell devices, where we get big improvements in being able to use a smaller cofactor, which has a higher diffusivity. We've done some other protein engineering work where we've tried to graft the activity of one enzyme onto this thermal stable scaffold. So we can take loops from a human enzyme and put them in our thermal stable enzyme, and we can see how that can give us alternative activities. More recently, we've done some really other sort of more exotic things. We've been able to take the enzyme and do directed evolution experiments to eliminate its enzymatic activity and actually turn it into a molecule that binds RDX, which is an explosive molecule, military explosive. And so we've been able to eliminate catalytic activity and turn it into an explosive binder for biosensor applications. And also more recently, we've been able to insert a domain in the enzyme. This peptide domain binds calcium. And so the interesting part about this is we now can put some steric stress on parts of the enzyme when we add calcium because of the folding of this domain, and that can influence whether the enzyme is able to use NAD or NADP. And we can toggle this preference back and forth by adding or removing calcium. And we've done a lot of kinetic characterization of this because it's an interesting phenotype. The next vignette I'll tell you about is also with this calcium-dependent peptide. We've done a lot of work with this beta roll domain. This is a peptide sequence that forms into this flattened corkscrew shape when we bind calcium atoms. And so what we've been able to do is characterize this using fluorescent proteins and by attaching it onto surfaces and exploring how the peptide folds and unfolds when you add or remove calcium. We've been able to rearrange and concatenate the different repeat sequences and to understand how that affects the folding mechanism. It turns out that there's a capping domain that's required for the peptide to fold properly. This adds then an entropic stabilization, enabling this folding mechanism to go from disordered to adopting this corkscrew shape. And if you don't have this capping group here, then it doesn't fold properly. Along the way, we were able to make some concatenations of these consensus sequences for these turns, and it turns out that precipitates and phase separates, which then is useful for purifying proteins with a non-chromatographic protein purification protocol, so we can use this to purify recombinant proteins. We've also been able to engineer one side of this beta roll domain to be very hydrophobic, and that allows us to also make biomaterials. So these are self-assembling protein hydrogels that only form in the presence of calcium. If you leave the calcium there, the hydrogel is pretty robust, but if we take the calcium away, the hydrogels dissolve away. And finally, we've been able to use directed evolution to engineer our beta roll peptides so that they now have affinity for target protein molecules. In this case, we evolved our beta roll peptide to bind lysozyme. And so now the peptide will bind lysozyme in the presence of calcium, but not in the absence of calcium. And this is now useful for affinity chromatography, where we can specifically purify lysozyme out of a protein mixture in the presence of calcium, and then we can elute it with calcium removal. So we went from sort of fundamental characterization of how the beta roll domain operates all the way to a technological application where we have a new approach for affinity chromatography. And the final story I'll tell you about is some more microbiology type work that we're doing, where we're interested in this bacterium here called Acidify-Bacillus ferroxidans. This is an interesting bacteria that's able to oxidize inorganic materials, either iron or sulfur, and it's able to fix CO2 and grow at low pH. It lives in sulfuric acid at like pH 2. We've been able to better understand and characterize its growth on different metals, and adding chelators to help prevent the inhibition by ferric iron as it oxidizes the ferrous iron. We've been able to grow the cells with vanadium as an electron donor. We've been able to do a bunch of genetic modification of this. We've established new promoter sequences to drive exogenous gene expression. We've been able to integrate foreign genes into the chromosome of the bacterium. We've been able to do genetic engineering of the bacterium, where we've been able to put foreign pathways in the bacterium to get the cells to now make biofuels from CO2. So we've put in foreign pathways and engineered the cells. And working with Professor West in the department, we've been developing electrofuels platforms where we can incorporate a bioreactor with our engineered cells and an electrochemical reactor, and combined, the electrochemical reactor can regenerate the iron and reduce the ferric iron back to ferrous. The cells can then oxidize the ferrous iron and make biofuels from CO2, and we can recycle the iron between the two reactors so that we're making biofuels from electricity and air. We've also had some interest in this project from the military, where we can try and understand how we could use the cells to start oxidizing metal resources as opportunities to extract energy from them. The cells are able to oxidize and to corrode some metals, but not stainless steel. We've developed a new mechanism where we can get the cells to be involved in the corrosion of stainless steel if we give them pyrite and chloride. And this allows them to make thiosulfate, and this is a new corrosion mechanism that can attack stainless steel. And this is ongoing work in the lab. So these are a couple of vignettes of things that we do, applied protein engineering for a whole bunch of different applications. This is my email address here. Here's our website. I'm happy to answer any questions. I'll be available to talk by email. And I look forward to meeting and speaking with you.